This is just a really quick overview of the assignments that you are going to have due for this unit. This is just a really quick write-up of the photosynthesis lab. So you're looking at the important components. What did you need to set this lab up? Why did those things ensure that this lab was A, going to measure photosynthesis, and B, going to create a successful experiment? And then in part B, you're discussing how did you choose to carry out the experiment? Mainly, what was your variable in part B? C, suggests 2 alternative variables and predict the results, so probably an if-then format. D, discuss the results of the experiment you designed, so talk about the positive control, what we expected to happen with the carbon dioxide. The negative control, the one where there was no baking soda, no carbon source, and then your variable. So you should be able to discuss the results of at least 3 different components. And then this part is about something called chromatography. So what it's asking you to do is try to analyze why does chromatography separate the pigments? How does it make sense that this liquid traveling up a paper can actually separate pigments based on their properties? And then you're analyzing, well, these are 5 different organisms, a bacterium, 3 protists, and a plant, such as spinach. So in this lab write-up, you're looking at what was the overall photosynthetic reaction. That's what we did with the floating disk. And then taking a second to look at pigments, which we know are really important to the light-dependent reactions. If we go to your analyzing photosynthesis, here you are supposed to define it. This is where you really need to get the electron transport chain and the chemiosmosis straight in your head. What are all the reactants? What are all of the steps, the redox reactions? What do I start with? What do I finish with? And you do need to know locations, like the thylakoid membrane, the thylakoid space, the stroma. What we have next is a primary source article that is about photokinesis and movement towards or away from light. And this is a behavior, and we actually see it in this organism, this algae. And so we're looking at how this algae actually uses ATP for its movement. And what is the source of that ATP? Because we actually know now that we make ATP in respiration. We also make it during photosynthesis. And so is there any way that this organism is moving because of what's being made, the ATP made in photosynthesis? Here you're creating a diagram of the different options. So the main option we know is the Calvin cycle, right? We know that the Calvin cycle will fix carbon, but we also know 2 other strategies to fix carbon. So we want to be able to describe the Calvin cycle, but we also want to be able to describe C4 plants and CAN and how they're different. And then this is actually looking at PEP carboxylase. So we just talked about PEP carboxylase, and now you're going back and kind of analyzing what is PEP carboxylase, and then reading a little bit about how do we regulate PEP carboxylase. After you finish working on your photosynthesis work, you definitely want to go back and analyze cellular respiration. Even if you've already finished this, you need to be able to go back and forth between one and the other. So being able to define glycolysis. Why is glycolysis so important? What happens in glycolysis? And then being able to talk about the ATP that is made during glycolysis and compare it with ATP made during oxidative phosphorylation. Then being able to look at, okay, well, what happens to pyruvate? We end glycolysis with pyruvate. What happens next? Turns out that we can do fermentation. We can oxidize pyruvate further. And you want to think about, why are we oxidizing pyruvate? Why are we fermenting? Why are these things happening? And then we've got a little diagram about your electron carriers, and you're trying to elaborate what you see happening here to our electron carriers. and then looking actually at the importance of those electron carriers and why are those electron carriers important. And so one final thing is that your test tomorrow is going to be multiple choice. You're actually going to have 2 full essays instead of 3 mini and 1 full. And what's going to happen is that you're going to write a full essay and then just bullet point outline another essay. And one of them you'll do in class and one of them you'll take home. The one you do in class, no notes, no nothing. The one you take home, notes, but no computer, please. Please try to really only use your notes. Last time, a lot of you, when I let you take the essay home, I could tell you did it maybe at the last minute, you took like 5 minutes. Treat it like it's an essay in class that you would take 15 to 20 minutes to write. And I would encourage you to really plan before you write. And please make sure you don't work with a partner. Some of you in your last essays, I felt like some things were a little similar, some little alarm bells were going off for me. So try to ensure that that's not going to happen again this time. And so this should help you get you ready for your test